Are you able to share your slides? I'll do a brief intro as Kym puts up her slides. Yeah, Kym is a PhD student, or she submitted her PhD very recently, and she's supervised by Ann-Maree Vallence, Hak Fujiyama, and Welber. And her research is some really interesting work looking at effects of tACS, transcranial alternating current stimulation on sensorimotor neural oscillations. So I'll let Kym take the stage.
Thanks, Leanne. Can everyone see my screen? Okay. Yep. Great. Yeah. Thank you for organizing this meeting and for the opportunity to speak. I really appreciate it. So, yeah, I'm going to be sharing a bit of my PhD research. So we'll get into it. I also included an acknowledgement, but thank you, Leanne, for doing that. So, my research was all centered around voluntary movement, which is fundamental for carrying out daily activity and interacting with our environment. So the sensorimotor cortex, as we all know, is a brain region that's important for executing voluntary movement. Now, interestingly, sensorimotor neurons actually work together in rhythms called neural oscillations. And though we don't have a full understanding of neural oscillations, they're thought to facilitate efficient communication throughout the brain and the body. Now, there are 5 well-established frequency bands of neural oscillations, and my research focused on the beta band because evidence suggests that this band plays a particularly important role in performing a voluntary movement. I like to envision the beta band like this, maybe a little bit of insanity that came with my PhD. Now, the reason why the plays an important, or we think plays an important role, is because when we move, there's a very distinct and reliable pattern of beta activity around the sensorimotor cortex. So before and during movement, there's actually a decrease in beta power or a decrease in the strength of beta oscillations. This is thought to reflect the activation of motor areas for movement preparation and execution. And then following movement, there's actually an increase in beta power that does surpass baseline levels. And this is thought to reflect motor inhibition, stabilization, and the suppression of additional movement. So unfortunately, many individuals do struggle with movement-related issues, and these individuals do show abnormal beta power during movement. So for example, motor impairment in both Parkinson's disease and stroke are both associated with abnormal beta power. So what if there was a way that we could actually adjust sensorimotor beta power? Could this improve movement? So this is where tACS comes in. So tACS is a promising technique that might be able to modulate sensorimotor beta power in humans. So just briefly, in case anyone here isn't familiar with tACS, it's a safe, painless, and non-invasive brain stimulation technique. And in tACS, a weak electric current is applied through scalp electrodes, which passes through to interact with the underlying neurons. Now, how might tACS actually modulate sensorimotor beta power? Well, research in animals suggests that tACS can modulate neural oscillations through a process called entrainment. And entrainment refers to the synchronization of the internal oscillations to the external tACS current. So for example, if I applied tACS at a beta frequency of approximately 20 Hertz, tACS should then in theory cause more neurons to fire at that same frequency. So what does the evidence look like in human research? There is some evidence demonstrating that tACS can modulate beta power. However, most of these studies have focused only on resting state beta power. And though this is very valuable, we do need to understand the movement-specific effects as well. Now, the 2 studies that have examined movement-related beta power only analyzed movement execution, but not preparation or termination. And it is important to examine all three of these movement periods because the underlying brain states differ, and differences in brain states have been known to affect the tACS response. And lastly, results have been inconsistent, so ultimately the effect of beta tACS in humans is unclear. So while tACS is a very promising technique, we don't fully understand its effect on beta power in humans. So a better understanding may unlock the potential of tACS as an effective and reliable tool for enhancing voluntary movement. So my primary research aim in my thesis was to determine whether beta tACS could effectively modulate beta power in a sensorimotor region. And in doing this, I wanted to investigate, or I did investigate, resting state power and movement-related power. And as part of looking at movement-related power, I included all three movement periods, pre, movement, and post. I also, as another aim, wanted to determine whether the stimulation intensity affected the tACS-induced changes in beta power. And the reason for this is because there's simply a need for studies to systematically investigate this parameter so it can be optimized, because there haven't been many investigations into this at this stage. So I recruited 21 healthy young right-handed adults. Why healthy adults? Just because we want a better fundamental understanding of the tACS response before designing protocols for clinical populations. And why right-handed? It's just to account for cortical differences between right and left-handed individuals. So the outcome measure here was sensorimotor beta power measured by EEG, which I'm sure everyone here is somewhat familiar with. And I think I look particularly great in it. Thank you. All right, so this is what the experimental timeline looked like. So had some early experiments set up, and then pre-tACS measurement of EEG, which there was a resting state measure and a movement-related measure. So for the movement-related measure, I had participants perform self-paced presses into a force sensor that was attached to a mouse once every 10 seconds. And I did that for about 60 trials. Then after putting the tACS electrodes on, applied 20 minutes of beta tACS over left M1. And the electrode placement was based on some computational modeling that I performed prior. I just didn't include that today. Additionally, the stimulation was applied at one of three intensities, or sham stimulation was applied. Then after removing the tACS electrodes, we took 2 more measurements of EEG, both incorporating resting and movement-related measures, just to see if any effects occurred immediately after the stimulation, or as closest to immediately after as possible, and then whether that outlasted that first initial measurement period. So the first measurement period was about 10 to 20 minutes post-tACS because of the time that it took to get the electrodes off and to reduce impedance. And then once again at about 30 to 40 minutes post. And then all participants attended 4 sessions, so it was within the subject's design. All right, so for the results. So did any stimulation intensity modulate resting state beta power? No. So relative to sham, none of the stimulation intensities significantly modulated beta power. So if we look over here and see that there are significant changes from pre to post following some of the real stimulation intensities, but none of those actually significantly differed from the increase that was also observed following sham. So why was there an increase following sham at all? Not entirely sure. Could be spontaneous fluctuations, could be a fatigue-related effect, which we've seen sometimes in the literature be related to a change in power, some sort of placebo effect. Yeah, not really sure. Now, why didn't any of the real intensities modulate beta power? Again, not really sure, but a few potential reasons. The parameters might not have been effective, so maybe these intensity, like the combination of intensity and frequency, might not have been effective to modulate beta power. It also might have been that EEG might not have captured the effect for a couple of reasons. One is that, you know, there could have been changes that were happening at like the individual kind of neuron level, but these might not be seen at a population level because some of the animal research has shown relatively, I think there was one instance where there were only like 9 neurons who showed a change in entrainment relative to like a much larger population of like at least 30. So not all neurons show entrainment effects, and so perhaps at the EEG level, which is looking at you know, relying on bigger changes in populations of neurons, it just might not have captured that kind of effect. And then alternatively, we took measures pre and post rather than during tACS because we couldn't get around the issue of the artifact that would have been induced in the EEG data. So it's possible that something was happening during tACS but then didn't outlast. And also it's possible that we did see some individual variability, and that, you know, when grouping the results together, that overall saw null effect. So in terms of EEG might not capture the effect because of the issue of not recording during stimulation. As part of my thesis, I wrote a journal club article on a novel protocol for indirectly evaluating entrainment during tACS. So as a commentary on this paper, just here, where they actually try to look at entrainment through EMG responses during stimulation. They didn't see any significant effects, but it's possible that it was due to some limitations. It's a promising approach. I'm not getting into it right now, but I just wanted to mention that there are some novel approaches that are being developed, which is cool. And if you want to read further, read my paper. Okay. All right. So I just talked about resting state beta power, but what about movement related? So interestingly, there were no changes specific to the beta band, but instead there were significant intensity dependent changes in a broader band of power, which was from 4 to 90 hertz. And this was specific to the pre and the post movement periods. So to try and break this down, 1 milliampere stimulation in the pre-movement period actually induced a delayed increase in broadband power. There's all very small changes, of course, as you can see. Now, why is a delayed response? Potentially due to late plasticity-like mechanisms that have been observed in post-tACS corticospinal excitability. That's my best guess at the moment. Now, in the pre-movement period, there were no changes following 0.5 milliampere or 1.5 milliampere stimulation, so only following 1 milliampere. In the movement period, there was no changes following any of the stimulation intensities. Why did this differ from pre-movement? It might be because, as I mentioned earlier, there are different mechanisms underpinning each period that might have affected the susceptibility to the tACS response. Perhaps something about the active engagement in movement might have affected the susceptibility. Or perhaps, you know, when movement was initiated, it kind of overrode that any change. Then in the post-movement period, found that both 1 and 1.5 milliampere stimulation induced a delayed increase in power. Now, notably, there were no differences between the intensities and the amount of the increase they induced, indicating a sort of plateau. And this might be due to homeostatic mechanisms that have been observed in other non-invasive brain stimulation methods. Interestingly as well, 0.5 milliampere stimulation actually induced a delayed decrease in post-movement power. Why the decrease? There was an interesting study, an animal study, that found that when the current is too weak to override the endogenous rhythm, it might create a competition for control between the external tACS rhythm and the endogenous rhythm, which then suppresses the internal rhythm. So that might be what's going on here. Just to add to the results, was peak beta associated with a change in power post-tACS. This was something that I explored as well. So just to briefly explain, peak beta is the frequency within the beta band that naturally shows the greatest power. So peak beta is something that varies between individuals. And the question I wanted to ask was, does the similarity between peak beta and the tACS frequency of 20 hertz relate to the tACS response? So, imagine you have someone with a peak beta of 13 hertz, 18 hertz, and 25 hertz. Would we all expect them to respond the same to one fixed frequency of stimulation? So I actually found that there was a significant negative correlation only following 1 milliampere stimulation and only for the pre-movement and movement periods, affecting this kind of cluster in the alpha beta region. And this was also evident at both post 1, which is 10 to 20 minutes, as well as post 2, which is about 30 to 40 minutes. So what does a negative correlation mean? Well, when I looked into it, it seemed that individuals with a peak beta further below 20 hertz showed a greater increase in alpha and beta power, and vice versa. So what does that mean? I don't really know. Just that there seems to be some kind of complex interaction between brain state, stimulation intensity, and stimulation frequency for neuromodulation, which I'm sure everyone using this loves to hear, but it's complicated. So yeah. So just to summarize, because I know I covered a lot there, beta tACS could modulate power, but the effects were intensity dependent and state dependent. Largely, no changes in the resting state for any frequency, decrease following 0.5 milliampere stimulation in the pre-movement period, increases at pre-movement and post-movement following 1 milliampere, and an increase only post-movement following 1.5 milliampere. And then also noting that individual differences related to the changes in sensorimotor beta power. So after this, I also, well, as part of this, really, I also was just kind of exploring whether the changes in power following tACS were local to the stimulation site, or whether they, the effects extended beyond the region. So just out of curiosity, I was also examining changes in the unstimulated right M1 and actually found some significant neuromodulation. So I'm not going into this specifically, but this led to a research question that formed the next part of my thesis, which was, what if tACS actually modulated the connection between M1s by just stimulating the left, just the connection between the two alter? So that's what I'm going to go over now. So just as a bit of an explanation, further explanation for the rationale of looking at the effect of tACS on N1-M1 connectivity. So power is not the only metric of entrainment. So that was something that I had been talking about largely up until this point, which is really the strength of the neural oscillation. So more neurons firing at peak beta frequency, greater power, less neurons, less power. But connectivity can actually tell us whether brain regions are more or less aligned in their timing. So this could be missed by measuring power because changes in connectivity can occur while the same number of neurons are firing at the beta frequency. So essentially we can see changes in connectivity, but not power. So I wanted to explore this. And then what did the evidence look like? There were only 2 studies actually looking at M1, like changes in motor network connectivity, or really just like connectivity between an stimulated and unstimulated region, because a lot of tACS studies, when they look at connectivity, they're intentionally applying tACS at 2 different regions and trying to look at whether or not they are synchronizing, whereas in this case I was just stimulating one area and trying to look at the effect on stimulated area. So of these 2 studies, one found that the stimulated left M1 became uncoupled from other aspects of the motor network. And then another study found that there were no changes in connectivity. So limited research, a bit mixed, just wanted to kind of investigate it a bit further. So that's what I did. So the aim here was to determine whether beta tACS could effectively modulate M1 connectivity, looking at all the same stuff as before and using the same data. So same study, and here are the results. So did any stimulation intensity modulate resting state beta connectivity? Oh yeah, and just for anyone who is interested, the measure of connectivity I focused on was on IMCO, or imaginary coherence. So yes, interestingly, sham and 0.5 milliampere stimulation led to an increase in M1 connectivity. But 1 and 1.5 milliampere stimulation decreased connectivity. So another, like, interestingly, the 1 milliampere effect was at post 1, so it was more immediate, and then 1.5 milliampere effect was actually a bit more delayed. Now, why was there a decrease following the higher intensities? I think this might be that kind of reduced coupling effect that one of the other studies had found. So essentially, by using tACS to try and synchronize the endogenous oscillations to the stimulation, it may not necessarily have ongoing effects to the other unstimulated region, and instead kind of misaligns them from what it was at prior to tACS being applied. And unfortunately, it wasn't incorporated as part of this study as to what the functional kind of response to this is. Like, is that a positive thing or a negative thing in terms of like what we see in motor control? I'm unfortunately not sure based on this research. I think it might depend on the type of task, whether it's unimanual or bimanual, and what part of the body is being used. It might be functionally beneficial to kind of separate the two to try and I guess upregulate one hemisphere, or it might be that by applying that stimulation to the left M1, that the decrease in connectivity might create an issue in terms of functional effects. So don't know, need to investigate further, but I think it's an interesting response. Now, looking at the movement-related changes, this is a quick summary because there's not much going on. So this is like a significant result. So it's in the movement period at 1.5 milliampere stimulation. So none of the other changes from pre to post were significant. So did any stimulation intensity modulate movement-related connectivity? Not really. yeah, we saw a broadband suppression of M1 connectivity in the movement period following 1.5 milliampere stimulation. That was really it. I also looked at whether peak beta was associated with the change in, this is power, I copied the slide, obviously, connectivity. There was a significant positive correlation for following 1 and 1.5 milliampere stimulation at post 1. These are in different frequency bands, and I can't really tell you why. And the positive correlation, looks like this, where individuals with peak beta further below 20 hertz showed a greater decrease in IMCO following stimulation, and vice versa. So individuals with peak beta above showed a greater increase in IMCO. Now, at post, in the change from post 1 to post 2, there was actually a significant negative correlation just here following 1.5 milliampere stimulation. And I think that's just reflecting kind of the extent of change from baseline going back down to probably returning to baseline levels. Essentially, individuals peak beta further below 20 hertz showed a greater increase in IMCO and vice versa. So to try and summarize the two analyses together, this is what we see. So a little bit all over the place, but interestingly, beta tACS modulated power and connectivity, but these effects are intensity dependent and state dependent. So for the resting state, didn't really see any changes in power, but saw some intensity dependent effects in IMCO, and then some effects in power in the movement period, but less in IMCO. So what does this mean? May need to adjust protocols for state-specific responses. And a really important question is, what are the functional and behavioral implications? If I could go back in time, I would include something there. It's also interesting to note that the stimulation intensity affected the tACS response in a nonlinear manner. So tACS administration should take this nonlinear effect into account. More is not always better, I guess, in terms of the response. And there's a complex interaction between brain state, intensity, and frequency, which we love. So it might be important to consider personalized effective response. Do you still hear me okay? Something came up on the screen. Yep, you can still hear me okay. Okay, good. Okay. So just on that note, in terms of personalization, in addition to the correlations, I noted a theme of inter-individual variability in response to tACS in my data. This is just a visual demonstration. This is power and connectivity results for different real intensities in the resting state. You can just see that each individual line representing changes over time at the individual level. You can see that overall there's a lot going up and a lot going down. So they're all over the place at the individual level. So because of this, I think an important step is to further understand variability and personalization. It's not just an issue in my research. tACS and other transcranial electrical stimulation techniques have this issue, and it requires addressing if we can address it, because it limits the potential of all electrical stimulation. So as part of my thesis, I also reviewed and summarized the current state of the field regarding response variability and the optimization or personalization of tACS. I'm just going to cover that a little bit now to kind of wrap up. So this paper here has been published, and in this paper I aim to provide a clear perspective of where the literature currently stands on the factors that contribute to response variability, and the methods that are being used, in recent years to reduce response variability and also provide recommendations. So a recent review identified 3 classes of factors that contribute. There are stable factors like individual head anatomy, genetics, age, sex. State-based factors, so that can be things like hormonal fluctuations, intake of certain substances, as well as experimental factors like individuals' baseline capacity for change. So recent studies have focused on using simulation software quite a lot to understand how differences in head anatomy specifically influence the current flow, because the current flow has been associated with both neurophysiological and behavioral responses to tACS. So these findings suggest that the response variability might be minimized by personalizing protocols to individual head anatomy. And this is particularly relevant for populations with known variations in head anatomy that might influence current flow, like older adults, stroke survivors, people with major depressive disorder, etc. So there's been a growing emphasis on reducing response variability by individualizing tACS parameters, including the electrode montage, the frequency for methods like tACS, and even the implementation of closed-loop methods have been getting a little bit more attention. So just to quickly explain of each of these, the electrode montage can be individualized by adjusting where we put the electrodes, how many electrodes, the shape of the electrodes, the size of them. Individualization can occur through simulations of current flow, where head models of each person are generated from neuroimages. Yes, so individualizing the frequency can also be done for methods that target neural oscillations, and it can be further individualized by trying to set the frequency to something like the peak frequency within a functionally relevant frequency band identified with MEG or EEG. Additionally, the personalization of montage and frequency are individualized through like an open loop protocol where stimulation parameters are based on one singular measure taken prior to stimulation. And the problem here is that these measurements don't account for the dynamic nature of neurophysiology, as well as the changes that may occur in response to tACS. So in a closed loop protocol, tACS is applied with concurrent or intermittent measures of neurophysiology. For example, taking a measure of EEG power, which can then be used as instantaneous feedback to update stimulation parameters. So in terms of where we're at with the efficacy of using these methods to reduce variability, at this stage, the extent of neurophysiological and behavioral benefits are still unclear, because few studies have directly compared the effects of the individualized or the personalized protocol to the non-individualized and a sham protocol. And then of the few studies that have compared, most of the results are mixed. So sometimes individualized performs better, sometimes it doesn't. And these mixed results might depend on variations in methodology and differences in outcome measures. So overall, individualized or personalized protocols are promising, but they require more time and greater resources. So we need more research to really verify that these protocols are more effective than fixed or non-individualized protocols. So in terms of future directions, I think it's important to determine the efficacy of the stimulation, so more studies comparing the individualized versus non-individualized. Studies should also consider individualizing intensity with current flow simulations. So few studies have tried individualizing the intensity, so that would be important to consider as well. And also it might be valuable to try and examine responses in human and animal models with comparable protocols to provide a comprehensive understanding of the mechanisms underlying these effects. Because it's really important to justify the investment of resources for pursuing individualization. And it would be good to try and individualize all parameters in both open and closed loop protocols instead of just kind of one parameter at a time. We want to try and like harness the full potential of tACS and research and maximize full benefits. So just to summarize from my PhD research, beta tACS can modulate local and network activity, but these effects are dependent on brain state, intensity, and frequency, and to fully harness the potential of tACS in research and clinical applications, it's crucial to address response variability, which can significantly impact the reliability of outcomes. And individualized approaches show promise in reducing this variability to each individual's unique neural characteristics. And further research investigating the efficacy of individualization is strongly warranted, as it will lead to more reliable interventions and hopefully improve outcomes for diverse populations. That's it. Thank you. Thank you. Any questions?
Thank you so much, Kym. Let me just change the screen. I had a quick question actually. So you mentioned, the M1 finding, and the first thing that popped in my mind was the change in stroke-related interhemispheric inhibition. Have there been any? I'm sure there probably is a lot of work, so please enlighten me and summarize this work for me about beta or your frequency changes in stroke patients, and have there been any clinical work that looks at tACS and stroke that address this?
I think just in terms of just like generally the effect of tACS for stroke patients.
Yeah, tACS and interhemispheric. Yeah.
I'm not sure if interhemispheric. I know there are some studies that have looked at stroke a bit, because there is some abnormal, like those movement-related changes show some abnormalities, I think, but I'm not sure. I can't remember, I'm sorry, at the top of my head, if these protocol, like any tACS protocol, looked at a kind of single site application of tACS or whether or not they try to, look across hemispheres. Yeah, sorry, don't know. That's interesting to, think about.
I was going to ask if that's all right. So really comprehensive and great work. I was going to ask, when I saw the box plots or the the effects that you're seeing are relatively small relative to variability, and then obviously you show the the individual subject stuff, and it's both directions quite variable. It seems like almost all of the brain stimulation kind of work that I've seen tends to have that kind of effect, that you see that there's a lot of individual variability, and even the original theta burst TMS stuff and all those kind of things, and PAS, and I'm just wondering if what do we take from that? Like,
is it worth it?
Well, I mean, obviously, the potential to make a difference non-invasively in this way is worth pursuing to try and work out. And as you're doing, I mean, all of the things that you're talking about in terms of can we individualize it, and what can we understand the factors that determine what affects for some people and has others. But I guess the alternative hypothesis is that it doesn't have, that these things have very, very small effects in general, and that their individual brain states vary over time, and that what you see pretty much all the time when you're stimulating is noise. So that's an extreme negative outlook, but I don't know, how confident are we that we're not there?
I think I definitely appreciate and hear what you're saying, and sometimes I have had those thoughts myself. I think because it's really, I would say, maybe over the last like decade, maybe a little bit longer, that really we started kind of having a lot more research highlighting that variability. So I think that's really been coming to the forefront, and because that's only been really happening in the last decade, like they've really been, you know, kind of just been starting to try and make changes to see how can we address this issue. And it is a little bit harder to achieve, but I think I guess we're trying to figure out how we can work with this issue rather than just be like, variability, maybe it's not, maybe we can't do anything about this, like, let's just leave it. I think we're trying to be like, okay, what's going on here? When we've made some significant gains in what we understand about what contributes to it, and then that, that takes time, and then trying to test these individual things takes time. So I think it's just taking time, but I think the potential's there, and I think we have seen really significant effects of brain stimulation in various populations. And so I think it's worth pursuing and trying to figure out, I think. And I think as with, even more, I guess, clinically approved kind of brain stimulation techniques, like in deep brain stimulation, for example, we're not there, it's not perfect. And it has to be used in conjunction with other like therapeutic interventions. I don't think the brain stimulation was ever intended to be like a one-all kind of fix for addressing an issue, but something that could kind of be used in tandem to help improve outcomes. So I think the potential's there and it's worth pursuing. It's just a bit frustrating.
Certainly that. No, I agree with your point. I was being devil advocate,
No, it's
asking
fine.
a very hard question, but I think I agree with you. And I mean, even in the case of deep brain stimulation, clearly it has enough positive effects that people are willing to invest a lot of, but we still don't understand the fundamental mechanisms of how that works. And we have a lot better chance of recording and understanding in that context. And for me, one more comment before I shut up, but I think understanding the things that we're trying to measure to establish what effect we're having is crucial. Like, if you're measuring the old TMS stuff, if you measure an MEP, that's a very blunt, indication of what's going on in the cortex. In this case, you've got power in various states, which again, maybe there's something very important going on in the cortex, but it's not really apparent on those kind of generic and blunt outcomes. So.
Yeah, I definitely agree. Yeah.
Can I ask something, Kym?
Yes.
Very nice talk. Thank you. Very
Thank you.
interesting work. This is outside of any reading I've done for a long time, so bear with me. But just for the tACS literature, recently there's been a bit of a shift, I'm probably biased here, but to trying in motor cortex much stronger intensities. So it was really nice to see that you had a dose manipulation. But with things like motor learning and tACS, we've been going up to 4 milliamps, and we've been finding more robust and what appear to be replicable performance benefits if we go up to 4 milliamps. So I'm just wondering if you have an opinion about whether with tACS we might get round some of this variability by upping the dose quite considerably and using relatively strong protocols.
I think, yeah, I think that would be really interesting to look at. I think there's only one tACS study that's looked at a strong dose, 4 to 6 milliamps in like alpha activity, and they actually didn't find significant changes in power that outweighed 1 milliamp per stimulation. So that's just one study, right? So it's not entirely certain, whether that's just because of that, it's not worth considering. I think it's so hard to know. I definitely think, relative to a lot of that animal work is seeing these very clear effects in terms of like the effect, entrainment. They use higher relative doses of stimulation compared to what we're getting in humans. So part of me thinks that, yeah, when we might not be getting enough in humans, and that like theory suggests that intensity should be kind of recruiting more neurons for entrainment. So I think it's definitely worth investigating, and I think, yeah, it would have been nice to try to, I think it's important to investigate kind of a greater range of intensities. The range that I selected was just based on kind of what was reported at the time, and there was some lower ones and some higher ones, and it's kind of limited in how many I could test. So we just tried to choose one above and below what the normal intensity is, but maybe that was too restricted, and maybe like a greater range is needed to see. But we even saw some slight effects there, so it would be interesting to see whether they yeah, what goes on there. I do actually I was just remembering that there was a study that looked at tACS at like 1 and 2 milliamps and on corticospinal excitability, and they found again that kind of different thing about like resting state and then like agonist and antagonist contraction of a muscle, like all of those different states in terms of corticospinal excitability showed differential effects for different intensities. There wasn't like, yeah, 2 milliamps like always does the best response. It was kind of like, what are we looking at kind of dictated which intensity showed greater change in excitability. So that's something weird and to think about.
Yeah, I think my take would be it's going to be probably non-linear, right? And I
Yeah.
wonder if when we're dealing with the relatively low doses, which most of my work has done as well, we're finding these non-linear effects, but we might find something different at those top intensities.
Yeah,
so
absolutely.
yeah, it's just interesting, definitely.
It is interesting, yeah.
Another little left field one if you're interested, but so I'm also really interested in the direct sensory effects of these stimulations to the scalp. So obviously stimulating the skin, and particularly when you've got alternating currents, then the sensory motor loop or the sensory loop back to the sensory motor cortex from the skin afferents might be important. I was wondering if you've had a sense of if you stimulate the skin and then record a somatosensory evoked potential, what that time latency is. And I wonder if that might have an effect if you've got essentially two rhythms being introduced. One is from this, so then they're out of phase, that might cause a reduction in effect. Whereas if they're in phase, you might exacerbate the effect. So if you were able to stimulate at a frequency that matched the time delay from sensory afferents to the cortex, yeah, it's a weird idea, but it always occurs to me that, you know, we need to be, and even with TMS, we need to be careful that we're ascribing our effects to the direct effects on the brain rather than indirect effects on the brain.
But I guess like then what would you do? Like in a practical setting, would you try to, you know, use numbing cream? I guess like if
Yeah, it's one option, or you could like, I mean, I think that from that location, you're probably looking between 10 and 20 milliseconds of time to reach the cortex. So you might be able to, and it's not, may not be the rhythm that you want, but you might be able to choose a multiple of that time with your stimulation so that you're in phase. The two arrivals of input to the cortex, one direct from the stimulation and one indirect through the sensory, actually are in phase rather than not in phase.
it's interesting. Yeah. Yeah, that's something interesting to think about.
Because I suspect there'd be quite a big, like when it's direct, it's much less, more likely to habituate, and the input to the cortex is going to be less pronounced. But when it's phasic like this, I think you're going to get a really quite strong sensory feedback rhythm coming in. So.
Yeah, I wonder if that, it's again, as you were saying, like if that's enough of like an issue to warrant trying to work around it, or if it's, but this will be interesting to investigate. I know there have been some studies that have looked at the kind of sensory response a little bit, but I'm not sure in terms of so much the lag, like just more in terms of like, is tACS affecting the brain, or is it through that sensory input? I think a bit of both, to be quite honest. Yeah.
I don't know if I can ask. I was involved. Perhaps I shouldn't ask anything.
Well,
Ask
you
your
can
question.
ask
I need
what
to
you
get a-
want.
I need to get a.
No, okay, just talking about variability and things that we control for. So what was crossing my mind, it's kind of along the lines of what Tim was talking about is, it could be as well how the person is perceiving the stimulation at that time. For example, if we believe that, well, there is some sensation going on and there might be activation of locus coeruleus and release of neurochemicals across the whole brain, that might also add to the variability that you have as a response.
Yeah,
So I don't
and
know
it's
how we
trying
can
to figure
go
out, I don't know, it's trying to figure out what's worth like honing in on and what's like at a point kind of like, okay, do we just, you know, take a step back and be like, is it worth kind of dissecting this so much for like what we're really trying to achieve? I don't know.
Yeah.
But it's so complicated, and oh yeah, absolutely. It's not, you know, there's not just one effect that tACS is having. Like it's not having these singular effects on these singular outcome measures. It's just that how I'm trying to better understand its output, but it's too complex.
Yeah, adding to that, Leanne was showing me some results that people, they said, "Oh, okay, I believe I was in the sham condition, or I believe I was in the real stimulation condition," and that was actually impacting the results.
Yeah.
So that's
Yeah.
another thing.
I don't know if you remember, but
Yeah,
thing.
probably mentioned that in your thesis, and I should have
Yeah,
read.
I think I mentioned that in my thesis. There was like a trend towards people being able to guess the higher intensities because they generally felt more intense. So, but I don't know. Can someone's ability? I don't know. I didn't include that in the model, so I guess don't know if it affected the results, but
Yeah, well, yeah,
Can we
perhaps
change?
later when you're less busy, we can try to have
Yeah.
a look at that and
Yeah.
keep that in the model.
yeah.
Expectation. But yeah, from a clinical perspective, I guess, like from say, like in that study that Welber was talking about, where people are treatment-resistant depression, it doesn't matter, I guess. You know, even if they were in the sham condition and they do feel better, so and their scores are getting better, it may not matter. Maybe from a clinician's perspective. I wanted to ask, is it generally the case that effect sizes are bigger in clinical populations for these types of stimulation studies than in neurotypical populations? Is it the case, you know, where we look at variability, maybe neurotypical populations are really wide in their variability, like, and then maybe because people have some type of disease state that actually changes their variability to be narrower and thus effects are more prominent?
Yeah, I definitely think the capacity for change is different between the two populations, and that it's hard. It's like I think I don't know. This has been something I'm questioning. Like kind of as I was like finishing my thesis, I was like, kind of wish like this was on a clinical population because like I wonder how transferable these effects are. Like some of the trends in terms of the complication, in terms of the intensity and that stuff is important to know. So that's important. But in terms of having like a functional kind of or clinical outcome, how I don't know how transferable this would be because there are differences between, you know, the healthy brain and the unhealthy brain. So just got to do more research, I guess.
With tACS, have there been lots of like null results published in clinical populations as well? Because with tDCS, yeah, we're starting to see some of those null results coming out, which suggests that maybe it's not always the case that using a clinical population is more sensitive. Is it the same with tACS, or is it just less research on tACS, therefore we don't know?
I think I won't lie, like I was very, you know, in finishing my thesis recently, more honed in on the healthy research. From what I saw, you know, much fewer clinical research, and generally saw some sort of significant results. So yeah, there might be a bit of a bias there in terms of the publication. Yeah.
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 Do we have any questions from anyone else in the audience? Alex, Cara, Johan,
It's okay if not.
I'm running out of time.
yeah, we're running out of time, I think, anyway. But yeah, thank you so much for your time preparing this presentation. It's really cool body of work. I will send around the recorded YouTube link soon. I haven't sent around the one to the last one. But yeah, I'll send that around soon, and feel free to reshare that so that you get more eyeballs on your research. Yeah, the whole goal of this is to get more publicity, I suppose, or have people be more aware of the type of work we do.
Yeah, I really appreciate the opportunity, and hopefully it was interesting. Okay,
No,
it's fine if it wasn't that very interesting. It's all very niche, right? But yeah, thank you.
all right, thank you everyone for coming. I really appreciate it.
Thanks, bye.
Thank you.
Thank you. Thanks.
Thanks, Kym.
Okay, bye now.